And I think we'll probably go ahead and jump right in. So I'm going to give it over to Michael Ford first from MS Bioworks. Like I said, we partnered with them for this webinar, and he's the first guest speaker up. So with that, I will stop sharing and take it away, Michael.
Good morning, everybody. Thank you for your time. Here we go. It wouldn't be an MS Bioworks presentation if I didn't at least plug the company somewhat. So just a little bit about us, what we do. We're a service organization based in the proteomics space. We've been doing this since, as MS Bioworks since 2010. And we've been working with proteome software for a long time, and we've been using the Scaffold products for a long time. And we're pretty excited to see this Scaffold Dia product come to market. I'm starting with an idea about what we've been doing and how it evolved and where we are now, and just going back a long, long time, relatively speaking, for some of you, with a look at the literature and how it's evolved and where we are now, from sequencing on triple quads through digesting with trypsin and present-day single-cell-based proteomics we're seeing come out, and distilling that down into a workflow that presumably a lot of people on the call will be familiar with and is really applicable to everything we do in the lab in one form or another, whether it involves fractionation or labeling or enrichment. These nodes are fundamental in the workflow. And if we look at how that workflow then is superimposed into the mass spec lab, and we look at the evolution of the hardware that supported what I'm calling an Orbitrap-centric workflow, we see that back in 2010, it was the Orbitrap Velos Pro, Orbitrap Velos Pro, with a linear ion trap and the D10 Orbi, and how that's moved through to present day, where now we're operating on in part on the Explorers 480, nanoscale chromatography, in terms of robustness, ease of use. We've seen a lot of technology come and go, but I think now we're at a place where we're happy doing UPLC coupled to these instruments. And then if we consider data-dependent proteomics specifically, we're looking again at the timeline of software and hardware evolution, quoting this from our perspective as a company, but then again...
Hey, Michael, sorry to interrupt. We're not seeing any slides. Are you sharing your screen?
Oh, yeah, hold on, sorry.
No problem. I just wanted to check if they were supposed to be seeing slides.
Oh, okay. It's sorry, I've got host disabled participant screen sharing.
Uh-oh, the host did something. That can't be me. All right, there we go. Sorry about that, everyone.
Okay, it's so nice. I'm going to show it twice. There you go. Okay, here we go. Is that good? We're good.
Yeah, it looks good now. Now we can see it. Thanks.
Cool. Cheers, Lindsay. Thanks. Okay, this is about the company, who we are, what we do, where we do it, and how long we've been doing it for. And it's pretty much industrial, academic, biotechnology clients, a lot of proteomics, and primarily data dependent. So this is the workflow I was describing that's evolved from the early days of some of the protein and peptide sequencing work to present day. And in one form or another, this is pretty much everything we do. And putting that in the context of the instrumentation, how the instrumentation's evolved, ease of use for nanoscale chromatography, robustness of the mass specs. Here we're going from essentially the Orbitrap Velos Pro through the Q Exactive HF, HFX, Fusion Lumos, and now Explorers 480. and they're all coupled with nanoscale chromatography in one form or another. And some of these are still in the lab, some of them have left the lab, but that's the way the field's moved from our perspective. On the software and hardware evolution side, hardware being computing power, obviously computing power has come down ridiculous amount. So when we first got started, we were running Mascot from Matrix Science, Scaffold from Proteome Software on the Beowulf cluster, and Dell workstations. And now we're running on PowerEdge servers with a lot more RAM and a lot more processing power, and really the cash investment there is not too crazy. But in this time, in this 10 years, we've seen some gains, and the gains have been primarily through advancements on the data processing side from Max Quant, to a certain extent adopting one unique peptide reporting through it, second peptide identifications through Max Quant, and then also match between runs. And those things combined have made label-free quantitation a little bit more attractive to customers. So with this increased sensitivity and robustness, that is less downtime on the instruments, the sensitivity gains have been pretty good. We've seen the high end, a 4-hour run, yielding something like 4,500 proteins. And this is with a QC we've been running for a very long time. It's a triptych digest from Promega. Promega helped MS Bioworks start up in 2010. And so you can order it from them. We inject 1 microgram, and we run this on the 2-hour run on all our instruments pretty regularly, every couple of weeks or so, independent of daily QCs, where that's just a BSA triptych digest. But really what you're seeing here is gains, incremental gains across a 10-year period. So in the background, as a service company, you know, we've been approached by a lot of people doing different types of things with data-independent acquisition in one form or another. And we knew that the folks at Proteome Software had a product, and that was evolving. And we were watching the space. And so the cartoons here essentially show the difference between the two acquisition methods, where in one, we're isolating a precursor, fragmenting it, and in the second, we're isolating a much bigger window and fragmenting everything. And so there is a computational challenge there that's been addressed by a bunch of folks, and there's some really cool stuff in the literature. And the paper I cite there is one that I found to be a really good read to understand the space. So we were looking out for opportunities to get into the space, and in 2018, we got the email from the ABRF. They do these kind of inter-lab studies that are really useful. And this one was for DIA. So we thought, okay, we'll get in here because they've got expert panelists and so on and so forth. And what we were looking for here is methods and how we were going to run things internally. So we're lab number 29. There's a paper associated with study that's on bioRxiv. You can get a bunch of information from there. But we were quite happy with the outcome, and we were impressed by the sensitivity. Just over 4,000 protein groups in 2 hours. So we thought, okay, that's pretty good. So we decided to explore it further. And then in 2020, when we had the lockdown, we were talking to the folks at Scaffold, and they said that they were really pushing the product now, that they were going to do a lot of development. Because this figure's from the manual. The Scaffold Dia manual, by the way, is absolutely phenomenal piece of work. It alone is worth reading. But this is one of the figures, and it's a pipeline for processing DIA data, and it's a little bit overwhelming. It can be overwhelming, and if you were to try and piece this pipeline together in your own lab from all your academic resources, it's not turnkey for sure. I'm sure in the right hands it would be, but if you're coming at this from a perspective of a greenhorn, it's not necessarily turnkey. So the DIA product then has essentially brought together all of these parts, absolutely amazing pieces of the puzzle, and put it all into one product, which runs on Windows. It's ridiculous. And everything that's here, I've put in the citations I could find, and I'm still we're still getting into the space, and so it's fun to be reading this stuff. But some of these products, some of these technologies are absolutely phenomenal. The Prosit, if you haven't sort of played around with it or even read up on that, worth a look. Just phenomenal. The encyclopedia paper, and then incorporating percolator and having it all available as one package is really phenomenal. Really, really phenomenal. So what we were particularly excited about was this development, and this was the idea of spectrum library-free approach to DIA, where we can use gas phase fractionation to make a library. And this is very attractive because from the experiments we've done, we've seen fantastic sensitivity with minimal overhead and really no concerns related to library generation. And again, this pipeline is made possible by Scaffold Dia in our lab. And I put a reference down there. Absolutely go read. Any references you see here, I've thoroughly enjoyed reading. So we're going back now to our sort of evolved pipeline. And we're taking a method from the literature and bringing it into our lab and reproducing it with no other changes beyond instrument settings and data processing. So we were very excited to do this. Excalibur now running on Windows 10. It has data acquisition options in there that can configure with reference to the literature. It's very easy to bring this on board. So this is kind of the punchline with reference to slides I'd shown earlier about incremental increases in sensitivity. This is as significant a gain in sensitivity we've seen in 10 years with millions of dollars invested in hardware. So that's a 30-minute run, and it's over 5,000 proteins identified in a 30-minute run. And so we've seen some stuff in the literature about biofluids, applicability there. That all looks very interesting, but just screening cell lines alone, this is a massive, massive opportunity. So in this particular example, then we wanted to show the software and the data acquisition method. We wanted to kind of put it in a position where we could really highlight the sensitivity and what it can be used for. So in this particular case, we have samples from gene-edited cells. And when people do gene editing, part of that pipeline that's highlighted here is selection of clones. So it's single clone culture and then some sort of assay to determine which cells have been edited. And a Western blot is a popular choice for that. And so obviously, depending on where you're sitting, Western blot is kind of like it's up for discussion, right? But if you can do targeted PRM, that's great. But yeah, the single cell culture and then some sort of quantitative assay to help guide cell selection. So we had 34 samples with 90 minutes per sample. And across the 34 samples, there was 6,100 proteins that are common. This is label-free quant. Absolutely, in the context of the workflow I showed, there was nothing here that needed doing to the samples after digestion. So digestion, solid phase extraction, onto the mass spec, and then 90 minutes and 6,100 proteins common, and a total of almost 6,500 proteins in 90-minute runs. And then this figure can be used to view which are the edited cells, which are the unedited cells, and then also to identify the edited proteins. And if you take because you've got so much data, right? you can do some pretty nice tests. And in this particular case, then we've got this t-test followed by hierarchical clustering analysis. And this is so much more depth than you would get from a Western. Absolutely ridiculous. And it's a 90-minute run per sample. So we're redoing this whole study now with much shorter runs to basically package into a day, a one-day experiment on a similar number of samples that can be used to guide some pretty significant decision-making, really. And this is just one example. So, you know, from the perspective of MS Bioworks and our involvement with Scaffold, proteome software, and now having Scaffold Dia online here, what do we see? We see quadrupled budgets for proteomic profiling. That's essentially the message. That before, where we would say we're going to do this experiment with a 4-hour profile, now we're going to use a 1-hour profile. And going back to 2010, where a deep profile experiment would be a 24-hour experiment on the Orbivelos Pro, to present time where we're doing a lot of proteomes with 4 hours of instrument time, with UPLC using data-dependent acquisition on the Fusion Lumos, one of the Fusion Lumoses in the lab, or what we just have the one Explorers 480 or on it. And no matter what size group you're talking to, people always want to do more experiments, and budget is always an issue. And so to offer people the opportunity to do 4 times as many experiments without changing the budget is huge. So, and we wouldn't have brought this online here if it wasn't for this product. So we are super excited about this product, super excited to have it in the lab, and really looking forward to bringing this workflow to market, really, and seeing where it goes. But it's a massive, massive advance, and we're appreciative of the work that the folks at Proteome Software have done here. And with that, I'll hand back over to Jacob.
All right. Michael, thank you for that presentation. We are obviously, of course, very excited about Scaffold Dia as well, and we're pleased and excited to see that you have found the results so beneficial and are looking forward to seeing some more stuff that you all are doing over there with DIA and getting some more feedback and everything. That's awesome.